and identification of genomic aberrations that cause diseases. And all numerical variants we can identify by cytogenetics, and whereas a few structural variants we can, with some limitations, we can identify by cytogenetics. So if you see, based upon the technologies, the modern human of cytogenetics is divided into three eras. So the era of golden era, from 1950 to 1960, you can call it as a golden era, because during this era they have introduced the methodology for getting the chromosomes. At first they experimented with various tissue. Earlier when they start cytogenetic, they started with bone marrow sample, and then slowly they have during this era they have come to a you know a conclusion that even the peripheral blood can be used for the analysis. And during that time what they used to do, they used to take the WBCs and Buffy code, and then they used to culture. Okay, so later in the second era, that is era of banding, which spans from 1970 to 80, there is lot of thing which has come out in this era, because the banding was developed in such a way that the chromosomes have become much more clearer for identification, for which there are lot of, because they are, the chromosomes are clear, even the microdeletion syndromes were identified, and some of the structural chromosomes were translocations or anything, all these things were identified during this era. And two basic things have happened during this era, that is the prenatal diagnosis, because everything was so clear they could offer prenatal diagnosis, as well as the cancer cytogenetics also had really prevailed in this era because of these particular microdeletions which were identified during this era. Then from 1918 till today, we call it as an era of molecular cytogenetics. So as morning Dr. Ashwin has told, that there is nothing like, now we can specify this is cytogenetics, this is molecular genetics. We ourselves do not know. It is an amalgam of both, and we call it as molecular cytogenetic. So this is a fusion of both cytogenetics as well as molecular testing. And majority of the tests which were developed during this era was FISH, fluorescence in situ hybridization, spectral karyotyping, and array CGH, okay, comparative genomic hybridization, and multiplex ligation dependent probe amplification, as well as next generation sequencing now is a good technology which is come out of this after this era. So what are the types of tissues we use for this culture? So peripheral blood is very commonly used. Even in cancer cytogenetic, we can use bone marrow, and amniotic fluid and chorionic villus for prenatal diagnosis, and skin biopsy to check for an alternate tissue. Suppose we wanted to see some mosaicism or something like that, then we can see the skin biopsies. And also products of conception can be used, where the tissues can be grown and we can see the anomalies which are there in the fetus. How do we prepare these chromosomes? So preparing the chromosomes, we can see chromosomes in the metaphase stage. So what is a metaphase stage? What is a metaphase stage? It is a stage which is present in mitosis. Okay, that means it's dividing. So what do we require? The primary requirement of cytogenetics is we require live cells. Okay, so anything dead cells, for example, there is a tissue which is frozen tissue or something, you can extract DNA. But for cytogenetics, it is not like that. You should have live cells so that you can grow that cell, and then you can see, you can get all the metaphase stage, and then you can analyze the chromosomes. Okay, so how do you do that? You take a peripheral blood and then you culture in a complete medium. So this medium, usually we culture the lymphocytes. We are interested in lymphocytes because mostly in the peripheral blood, around 40 to 50% are the circulating lymphocytes which are present, and we are going to capture, and out of which around 55 to 75% are the T cell T lymphocytes, and around 15 to 20% are B lymphocytes. But we are targeting T lymphocytes because we use something called as a phytohemagglutinin. Okay, so this is a mitogen which will target the T lymphocytes to proliferate. That means allowing the cell to divide. And then take this peripheral blood, we add medium and then PHA, and we culture it for 72 hours. So this is a short-term culture, 72 hours, because our cell cycle is around 18 hours. So almost like 3 or 4 cycles you finish when we get this metaphases. And after which you harvest the cells. So harvesting of the cells requires 3 steps. One is like it is mitotic arrest. So after 72 hours or just 1 hour before or 2 hours before that, you can add this colchicine or colcimine, which is an analog of colchicine. So what it does is it is a mitotic poison. So after metaphase, the other next stage is telophase. So then what happens in telophase? The metaphase, after metaphase, the spindle fibers will be formed, and these spindle fibers will be pulling towards the poles. So when we add colchicine, that will poison the spindle fibers, and spindle fibers will be dissolved, so it will not allow the sister chromatin to go on. Okay, so it will be retained in a metaphase stage, so we can capture more number of metaphase for our analysis. So that is the reason. Then the second step in harvesting is you are adding hypotonic solution. So what is hypotonic solution? Hypotonic solution. So what is hypotonic solution? So we are taking KCl, and then we are adding this hypotonic solution to the cell. What does, it's a simple osmosis from where from higher concentration the solution will enter into the lower concentration. So the cell which is present, that will become large. Okay, then cytolysis will happen and the cell will break, releasing the chromosome. So that is the function of hypotonic solution. The third one is we are fixing the cell. So once the chromosome comes out of the cell, then you are adding methanol and acetic acid, acetocarmine we call it, and you put it there, and then the chromosomes are fixed. Then you harvest, you give good washes, remove all the debris of the RBCs. Then you have only WBCs which are present. Give good washes so that there is no remnants which are present from the RBCs or any other artifacts which are there. Then you prepare slides, then you band or stain them, and these are the metaphases what you get. So the whole procedure of this takes almost 72 hours. So basically, and the other thing is, when you are collecting a sample for chromosomes, you should collect the sample in a green vacutainer, that is a heparin vacutainer. Okay, you should not collect in EDTA because it will interfere with the cell growth over here. So basically, if you see what is a chromosome structure, it is having a centromere followed by 2 arms. The arm which is shorter we call it as a P arm, and the arm which is longer we call it as a Q arm. And then the tips of the chromosomes are protected by something called as telomeres. So these are the different structure for chromosomes. Now, how do we classify the chromosomes? So classification of chromosomes is based upon 3 ways. One is based upon the position of the chromosome, size of the chromosome, and binding technique. So if you see, based upon the position of the chromosomes, we have 3 different type of chromosomes in humans. So the metaphase chromosome, where a centromere divides the chromosome into 2 equal half. And then submetacentric chromosome, the P arm is shorter when compared to the Q arm. And acrocentric chromosomes are the chromosomes where the P region will be having only satellites. So okay, these are the satellites which will anchor the ribosomal RNA. And then the Q arm will have a euchromatin material. So these are the 3 types of chromosomes. So classification, one of the classification can be done based upon whether they are metacentric chromosomes, submetacentric chromosomes, or acrocentric chromosomes. So the second type of classification is based upon the size of the chromosome. So based upon the size of the chromosomes, they are divided into 7 groups. They're from A to G. A, B, C, D, E, F, and G. So A comprises of 1 to 3, and B comprises of 4 and 5, and C group from 6 to 12, and D from 13, 14 and 15, and E 16, 17, 18, and F 19 and 20, and G 21 and 22, and sex chromosome. X is almost the size of the C group, and Y is little greater than F group. Okay, so this is how the classification. All this classification is done because just to identify the chromosomes in a easier or a better way. Okay. Now there are some exceptions because pre-genomic era chromosome 21 is smaller, but still it is 21, and 22 is larger than that because of the Down syndrome was discovered a little bit early when compared to the number of the chromosomes. But post-genomic era, we know that chromosome 10 is smaller than 11, and chromosome 12 is smaller than 10, and also 19 is smaller than 20. So these are the exception because at that time we could not see at the genomic level, but whereas now we know what is the size of each chromosome, that is why they have placed properly. And the third one, an important one, what we are using right now is based upon banding pattern. So ideally, and this is an ideogram, which is a diagrammatic representation of a karyotype. Okay, so GTG banding is G banding using trypsin and GIMSA. And based upon this banding, ISCN, which is an international system of cytogenomic nomenclature, has classified chromosomes from 1 to X and 22 and X and Y. So based upon this also, world over, we are reporting these cytogenetic abnormalities. Now let's see what is the description, how we give an address for a chromosome. So this is a chromosome. So each chromosome, as I told you, will have a P and a Q region. So first thing when you see a chromosome, you should search for a centromere. So this is a centromere, and this is a P region, and this is a Q region. So now each arm is divided into regions. So these regions are divided in terms of landmarks. Some landmarks are present on each chromosome, and these regions are divided, and these regions are divided into bands. So if you see, like we wanted to identify this 9q24.2. Okay, so you should never read out as 24.2. It is a 2, 4 and a 0.2. So here if you see, this is a green colored is a region. That is, here we are interested in chromosome 9. This is chromosome 9 ideogram, and Q. So you should come below the centromere, and then 2 is this region is 2. And then these regions are again divided into bands. So these are the bands, and what is that we are searching for? Fourth band. So you can see this is the fourth band over here, and these bands are further divided into sub-bands. This is a 0.2 band, here you can read out. That is what we are seeing for. So when this is important because identification of a chromosome or writing a report, everything will be dependent upon this position or the description of a chromosome. Now what is a common terminology? There are certain things which we should know. What is that? Mosaic. What is mosaic? An individual with 2 or more cell types differing in chromosome number or structure. Okay, so some cells will be 46XX, some cells will be 46XY, or whatever it is. In case of Downs, you will have normal cells as well as you have a trisomy 21 cells. Marker chromosome. So in cytogenetics, anything which we cannot identify, which is the size of a chromosome 19 or 20, that we will call it as a marker chromosome. And satellites, as I told you, satellites are the P region of the acrocentric chromosomes. And importantly, we should also know what is a derivative chromosome. What is a derivative chromosome? Any chromosome which undergoes structural changes due to any chromosomal rearrangement like deletions, duplications, or anything translocations. So that is called as a derivative chromosome. So this is the basic terminology what will be used in cytogenetics so that you can converse with about, okay, this is a case of a derivative chromosome, or in this karyotype we have a marker chromosome likewise. And all these can be referred in ISUN 2022. Now the new one also has come. So what are the type of banding? So basically there are several types of banding, but these are the common things which we can report regularly. That is GTG banding, C banding, NOR staining, and DAPI staining. So GTG banding, as I told you, G banding using trypsin and GIMSA. So we know that chromosome is a structure which is made up of histone proteins, and the DNA is found along these histone proteins. Because morning, if you have heard Dr. Yatish saying that he works with chromosome architecture, and he was telling how this 2-meter DNA is compacted within the cell nucleus. So we all know that chromosomes are present inside the nucleus, okay, and all of them are present as 46 chromosomes. So 46 DNA molecules are wound around these histoproteins. Now what trypsin does is, trypsin is an enzyme which will, endolytic enzyme, which will extract the histones. So leaving the skeleton of the DNA like that. And this, then we add a stain called GIMSA, which is a complex stain, and this stain will go and sit in the AT rich region. Exact mechanism we do not know, but it is observed that it will go and sit in AT rich regions, and wherever the AT rich regions are present, it will be darkly stained. And these regions are the heterochromatin region, and wherever the light regions are, GC rich regions are there, it will be lightly stained, and you can find as light color band. So if you see GIMSA positive, then it is called as heterochromatin band, and GIMSA negative we call as euchromatin region. Okay, that is how you can. And as I told you that the era of banding has really revolutionized the cytogenetic field. Earlier they were just doing the staining. Okay, so you can see only these chromosomes, and you cannot really align the homologous pair of chromosome. But whereas here you can see after banding, it is so clear, you know what are the landmark bands which are there. Based upon the landmark bands, you can align both the homologous pair of chromosomes. Now what we can identify by using this GIMSA staining? So we can identify numerical abnormalities like trisomies, monosomies, triploidy, tetraploidy, and also structural chromosomal abnormalities like intrachromosomal abnormalities like inversion, ring chromosome, deletions, duplications, and isochromosomes. And interchromosomal abnormalities like reciprocal translocation, that means okay, let me tell in detail, and then Robertsonian translocation. So these are all the anomalies where we can identify by doing a simple GTG banding. So the first one is trisomy. Somy is an increase in chromosome number. So trisomy is aneuploidies. All aneuploidies you can identify. This is a trisomy 21, where an additional chromosome 21 is present, and these originate because of a meiotic non-disjunction and abnormal meiotic recombination. So you can see triploidy 21, you can see 13 as well as trisomy 18 also. Monosomy is the absence of a single chromosome. They are very rare and they will be eliminated early in gestation. The only viable monosomy is Turner syndrome. Only one X chromosome, because females can survive with one X chromosomes. So this happens because of anaphase lag which happens. So one X will not enter into the anaphase, where then cytokinesis occur without the chromosome, and in one cell it will be 45 XO, and the other one there will be only 45 X, 46 XX will be present. And triploidy. Ploidy is an increase of whole set of chromosomes. So here if you see like additional whole set of chromosomes, making it 46 plus 23, 69 chromosomes. And these we can see only very common in miscarriages, and it happens because of the failure of meiotic division, resulting in a gametes with 46 chromosome with a normal sperm, or vice versa. In tetraploidy, two sets of chromosomes will be present, because there will be a failure of an early mitotic cleavage division. So instead of the cell cleaving, it will not cleave, and then they remain to be in one cell, making it for 92 chromosomes. But again, this is not viable. You can only see them in aborted materials. So interchromosomal are reciprocal translocations. So reciprocal translocations are a two-way exchange of the genetic material between two non-homologous chromosomes. So you can see here some part of chromosome, this is normal chromosome 2 and the other chromosome is normal chromosome 5. But you can see that there is a reciprocal translocation which has happened from chromosome 2 to 5 and vice versa. So likewise, there are lot of translocations which can take place constitutively from chromosome 1 to 22 and X and Y also. And these are mostly in constitutive cytogenetics, they are non-recurrent. Unlike in cancer, they are quite recurrent. Like you can see in AML or CML or any hematological malignancies, you can see BCR-ABL, which is a very common thing. So likewise, we will not have very recurrent translocation, they will be non-recurrent, and our interest is like anything which is non-recurrent and associated with the disease, it will lead to a phenotype where you can work on that particular case. And interchromosomal translocation, again I told you, there is another type of translocation called as Robertsonian translocation. So this is a fusion of two acrocentric chromosomes. So in humans we have 5 pairs of acrocentric chromosome 13, 14, 15 and 21 and 22. So anything which is happening between these 5 pairs of chromosomes, we call it as a Robertsonian translocation. So here you can see a Robertsonian translocation which is happening between chromosome 13 and 14. But this is 45 chromosomes only, but still this carrier of this translocation will be normal. Only the problem comes when there is a problem in meiosis for his offspring. Okay. And the intrachromosomal, anything which happens within the same chromosome, those are called as intrachromosomal translocation. And one is inversion. So inversions happen when a break occurs on a chromosome, on either side of a chromosome, and then the segment which is present between the broken region will be twisted to 180 degrees, and then again it will be attached. So if you see this is a normal 3 and this is also a normal 3, and you can see this is inverted here. Okay, so if it is involving a centromere, we call it as a paracentric inversion, and if it is not involving a centromere, then we call it as a pericentric inversion. So this is an example of an inversion 3. So ring chromosomes. So I told you the chromosomes are always capped by centromeres. So why they are capped? Because there will be, if they are not capped, then they form sticky ends, and then they can go and interact with any other chromosome, forming the aberrations. See, that is what happens in cancers, where the telomeres will be shortened, forming sticky ends, and there will be a lot of aberrations, translocation, deletion, duplication, all possible things occurring in cancer thing. So here in ring chromosome formation, the telomeres will be deleted on either side, forming a sticky ends, and then they again rejoin as a ring. The whole thing will rejoin as a ring. So based upon the extent of the material which is gone, the ring, if the ring is bigger, just below the telomere, if there is a break, bigger ring, then probably he could be normal. And if there is a larger part which is deleted, a small ring is forming, that means major chromosomal region is being gone, so that leads to a disease. So most of the people with the ring chromosomes have seizure disorders. Okay. And duplication. So duplication is an addition of a part of a chromosome or arm of a chromosome. Anything can happen. So anything which is having an extra, you can see this is a normal 5 and this is an extra chromosomal material which is coming as a tandem duplication. So this is called as duplication. So duplications can be tandem, that means which are present just behind each other, or it can be somewhere else also. This part will be duplicated, where we can identify by some other techniques. Deletions are the loss of any part of a chromosome, part or a band or an arm of a chromosome. And here this is an example of a deletion, XQ region. So this is a variant of Turner's syndrome. Usually if there is a bigger deletion like this which is happening in autosomes, it will not survive because there will be so much of imbalance with the genome. It either leads to a phenotype or it will be aborted. It will not come into term at all. So isochromosomes, this isochromosome is a chromosome where one of the arm of the chromosome will be lost and the remaining arm will form a mirror image. So here if you see, this is an isochromosome X, and mostly common isochromosomes are with chromosome X and Y. And here this is a normal X chromosome and this is a centromere, and this is also a centromere for the iso X. You can see that there is a Q region which is there, and this Q region will be forming a mirror image, and you can see Q again on top of this instead of P. So what happens here, there is a monosomy of this P region and there is a trisomy of this Q region, 1Q, second Q and 3Q. So this is also a variant of Turner syndrome. You imagine the same thing happening between autosomes, so it is not possible. Okay, so it doesn't come into terms. So as far as I seen in my whole career in cytogenetics, I have never seen any isochromosome formation with any of the autosomes. So the second type of banding is C banding. This is a centromere banding. So we have some constitutive heterochromatin regions present in our cells. They are present on chromosome 1, 9, 16 and Y. So if there are any anomalies which are present, in this region, suppose there is a bigger chromosome 1 which we are seeing, then immediately we can do this particular staining, and you can check whether there is a centromeric region which is extra in this region or it is a euchromatic material. Okay, so quickly, only on these 4 chromosomes you can do this particular staining. So here is a clinical importance which I will show you. So this is a case who has come to us with recurrent miscarriages in wife, and then the karyotype in the husband showed that there is an additional material which we have found in this 16. So ideally when we see he's normal, because the only problem is they have come for as a BOH case, and then we know either 2 possibilities could be there. There could be a cryptic translocation which we cannot identify here, or then we have to identify what is this, whether this is a true duplication or something like that. Then we have done C banding because this is chromosome 16 where there is a constitutive heterochromatin, and you can see a bigger chunk of this heterochromatin region which is stained. So we can say that it is a normal polymorphic variant which is present in this particular case. And NOR staining is one nucleolar organizing region staining. As I told you, we have 5 pairs of acrocentric chromosomes. So all these acrocentric chromosomes, I told, they are satellites, and these satellites are stained with silver nitrate solution. So when silver nitrate goes and stains here, it becomes a dark black dot-like structures, and you can see here. This is a case where we had a extra chromosomal region which is present on chromosome 13. So anything 13, 14, 15, 21 and 22, we go ahead and we do this AGNOR staining. So this is a non-consanguineous couple who has come with a bad obstetric history with 3 miscarriages, and the wife showed 46XX normal, but the husband has this. So we have done a simple AGNOR staining, but what we expect that if there are dots, that means it is a normal polymorphic variant. But here in this case, what we have identified is, we have seen here this is derivative 13. So I am using the word derivative because it is an additional material which is come from some other region. So here they are not stained. The P region usually has to be stained, but they are not stained. But to our surprise, we have seen that it is these acrocentric or satellites, they are present on the Q region of one of the submetacentric chromosomes. So we could identify that this is a translocation which is which is found between chromosome 11 and 13. So this is a cryptic translocation which we could not identify by cytogenetics. So because of the resolution of cytogenetics, we could not identify. So even a simple techniques which no labs will do, because they think that this is not at all an important technique. But this, because of this simple technique, we could really identify that there is a translocation. So next time when they come for prenatal, we also have done prenatal, and then we have checked for the translocation. They had a normal baby. So the other staining is dapping. This is a fluorescent stain which will stain the nucleic acid stain. So the reason why we do this quickly with a fluorescent, but the thing is you should have a fluorescent microscope, because it will be only seen with under the microscope, this fluorescent stain. So this is if you see, this is a case which has come to us with a small y and a translocation. On an extra material which is found on chromosome 15. So usually we do it in two cases, where we can check and quickly Y and then autosomal translocation which we are suspecting. So here, because there is a small y, there is an additional material here. We wanted to check how the staining will occur, and then we could identify that most of the thing is going and sitting on this chromosome 15. I didn't put the picture over here. This is a normal, this is a Y of this particular case. And the other case here is, there is usually the Y size will be little greater than 21 chromosomes. So this is a big Y, but it can range from chromosome 15, the bigger one. So usually in these cases we think that there will be a translocations or something which we will be suspecting. But here, when you do simple, you can see that there is a bulb-like structure which is present, indicating that it is a heterochromatin region which is present. So it is a normal polymorphic variant here. So if you see, I'll end up with this. This is a genetic counseling thing which I want to put this Robertsonian translocation. So this is a case of 45 chromosomes which are present. Okay, you can see a Robertsonian translocation which is happening between chromosome 13 and 15. So the chromosome number is only 45, but the genome is balanced here. The carriers are normal, but the risk of reproductive issue will be there due to abnormal segregation of the chromosomes during meiosis. So here, 45. So in here, what we do, the risk will be only like you can tell to the couple that they can come for a prenatal diagnosis, and you can do the risk will be like what could be the risk in this case. Can anyone tell? Actually, I wanted to ask you guys, what could be the risk if it is only 45 chromosome with a Robertsonian translocation involving only D group? Yeah, someone is telling. Rupa? Yes. Okay. What is the risk? And it is very easy to handle this case, because you can do a prenatal diagnosis and you can check whether there are any trisomies. Usually in such cases, if there is a trisomy or a monosomy, it will not usually come to term. If it is coming to term, it could be either a balanced translocation. Ideally, it could be a balanced translocation, or a normal gametes might have come. And the second thing here, there are two conditions which I would like to show. So this is a translocation which is happening between chromosome 14 and chromosome 21. So this is a carrier. His father is a carrier. And the other one is chromosome 21 and 21. So what could be the risk in this case, and what could be the risk in this case?
For Down syndrome.
Yeah, for the Down syndrome child. Sorry.
On the right side, you will 100%.
Correct. The left side, will it be same as this one? So 10% if a mother is a carrier. So in this case, it is a father. So around lesser than 5%, this is a risk. And here in this, it is a 100% risk. The reason why I showed this is in all these three cases, it is a Robertsonian translocation. But how you should go and counsel is different. So you should always know with what are the chromosomes which are involved, because that also will give you an insights how to go ahead for counseling. Okay. So if there is a book written by me. If you want, you can just go through the book, and anyone are interested, you can get back to me. So I would like to acknowledge our core grant, CDFD core grant and project, our mission program, and Dr. Ashwin Dalal for his support, Uzair and Vijay who are there, and patients and their families. Thank you very much. Any questions? I can take.